the endocannabinoid system, and cannabidiol, CBD. The endocannabinoid system is a lipid-derived signaling system discovered within the past decade. Cannabinoids, which are homeostatic regulators, circulate throughout human and animal systems continuously, affecting all physiological processes. The endocannabinoid system is comprised of CB1 and CB2 receptors, which bind directly or indirectly to cannabinoids and phytocannabinoids. CB1 receptors are excitatory and are located in the central nervous system, lungs, liver, and kidney. CB2 receptors regulate immunological responses and are located in the immune and circulatory systems. Endogenous compounds, such as anandamide and 2-arachidonoglycerol, are made by mammals from lipids and bind directly to the CB1 and CB2 receptors, serving as neurotransmitters for cannabinoids. Cannabidiol, CBD, a non-psychotropic cannabinoid naturally occurring in human and animal species, is also a phytocannabinoid derived from the industrial hemp plant. While CBD does not bind directly with receptors, it does affect stress genes such as SO8-2 and CYP27A1, which controls sterol-like cholesterol metabolism. CBD increases the amount of anandamide and other vital lipids, thereby indirectly increasing the availability of circulating cannabinoids to bind with CB1 and CB2 receptors. Research has shown that cannabidiol, in the form of CBD oil, has therapeutic benefits individually and adjunctively with other interventions. Cannabidiol, made from legal industrial hemp, contains less than 0.3% THC, rendering it non-psychoactive. CBD oil has anti-emetic, antioxylytic, anti-tumoral, and immunologically inhibitory properties. 3 categories differentiate the types of clinical endocannabinoid deficiency, which are associated with different disease processes and disorders: genetic, acquired, and idiopathic autoimmune. Many disorders have a combination of CE/CD origins, and supplementation with CBD cannabidiol requires ongoing assessment to facilitate optimal benefit for the individual. The human endocannabinoid system is responsible for memory networks in the brain, both excitatory and inhibitory, including the neurogenesis of hippocampal granule cells, which regulate the timing of the endocannabinoids in accordance with the brain's needs, pain perception, mood, appetite and taste regulation, and metabolic function, which regulates the storage of energy and transport of cellular nutrition. The endocannabinoid system affects the lipocytes and fat cells, collectively known as adipocytes, hepatocytes, in the gastrointestinal tract, the musculoskeletal system, and the endocrine system. Cannabinoid receptors CB1 and CB2 facilitate the responses of the endocannabinoid system in the body, which are critical to maintaining homeostasis. CB1 receptors are located in the central and peripheral nervous systems, as well as the lungs, kidney, and liver. CB2 receptors are predominantly expressed in the immune system and hematopoietic cells. Because some phytocannabinoids, cannabinoids of the cannabis plant, and hemp cannabidiol mimic components of the endocannabinoid system of the human body, the effects are of interest in the medical management of multiple disorders, including disorders of the ocular system, which is directly affected by the immunological and cardiovascular systems. Indeed, research indicates that both THC and cannabidiol CBD have neuroprotective and therapeutic ocular benefits. Cannabidiol is a non-psychotropic and non-toxic compound which has been demonstrated to positively affect the human endocannabinoid system. Cannabidiol, derived from the hemp plant, demonstrates anti-inflammatory and immune-modulating properties. Cannabidiol has a low affinity for CB1 and CB2 receptors in the human body, but acts as an indirect antagonist of their agonists. Antagonists are defined as substances that stop or inhibit the effects of another substance on the cellular surface, producing the same effect as a substance which would normally bind to the receptor. Agonists are chemicals that bind to the receptors and elicit a biological response. Therefore, cannabidiol may enhance the therapeutic effects of THC, possibly by increasing the density of CB1 receptors. Cannabidiol has been demonstrated to cross the blood-brain barrier and exert antioxidant, antimicrobial, and neuroprotective properties, rendering it valuable in the prevention and treatment of oxidative ocular disorders and diseases.